Start.
So class 12, the first practical is that is dissection of Hibiscus rosa-sinensis. You all know Hibiscus rosa-sinensis is জবা ফুল যেটা প্রত্যেকটা বাড়িতে পাওয়া যায়, family is Malvasia. 4 holes থাকে, that is 4 holes we have all the flowers, most of the flowers, and Hibiscus rosa-sinensis advantage is that is 4 holes are actually present in all the flowers. First hole is calyx, second hole is corolla, third hole is androecium, and fourth hole is gynoecium. So first hole that is comprised of sepals. Sepals বাইরের যে green কালারের পোরশনটা থাকে, green কালারের যে পোরশনটা থাকে এগুলোকে আমরা sepals বলি, but here one extra part I have written, that is epicalyx. That part is this, all these things. One extra outside holes are present. Outside of calyx is present. Calyx is this green portion outside of the flower, and one more outside part is there, that is epicalyx, the outside part of calyx. So one more part, that's why here 5 parts I have written. So first holes, again I am repeating, first holes that is sepals, this portion, this fused portion called epicalyx. So here in Hibiscus rosa-sinensis, one more extra outside part is there, that is epicalyx. So for this you can use your finger to pluck it. You will get dissection tray whenever your practical will be there. I am doing on a page to make you understand more. So this is epicalyx. This is actually the extra outside part of calyx. So next we are coming to the first holes, that is sepals, which is collectively called biological word calyx. So this is the, so here you have to dissect so that you can pluck it. See that it should be joined. Most of the flowers it is 4 to 5, most of the flowers it is 4, 5 or more than that, 8 also sometimes there, but here maybe 6 or 5. Let me count how many are there. 1, 2, 3, 4, 5. So here it is 5. This is called calyx, and it is all are fused together, and fused calyx are known as gamosepalous. You have read in the book in class 10 also, 8 also, that all the fused calyx are known as gamosepalous. So all are fused, একে অপরের সঙ্গে লেগে আছে. So the calyx is over, that is first hole is over. So next second hole is corolla. Corolla is all of you know very well, that is the petals of the flower. It's sometimes 4, sometimes 5, and so on it goes. So second hole is petals and collectively called corolla. That is, you can use your fingers to pluck it out, all the petals. Here it is 5. So this is corolla done. Now androecium, that is the male part of the flower. This is actually androecium, this one, that is the staminal tube. This one is staminal tube, that is pollen tube. These are all pollen grains. These yellow like structures are pollen grains, powdery like structures. This is staminal tube, and it is the male part of the flower, that is androecium. Now from here we have to dissect the staminal tube to bring out the female part, that is gynoecium. Gynoecium consists of 3 parts, that is stigma, style, ovary. Stigma are these things, these red color things are stigma. This is also 5 in numbers, I think so. Yes, 5 in numbers. 1, 2, 3, 4, 5. This is stigma, that is sticky like substance jar opore insects ra eshe boshe, and the pollen grains used to stick on the insects so that cross pollination takes place. So now the inside portion, that is style, and this is the swollen part, একটু ফোলা মতন জায়গাটা হচ্ছে ওভারি. So the tougher most part is we have to dissect it. And you have to do it very carefully so that the inside portion, that is style portion, do not get torn out. Yes, this is the white portion that you have to bring it very carefully. Yes, this one. This one is gynoecium. So this is stigma. This long tube like structure is style, and this is ovary, that is the female part, and this one is actually the androecium, that is the male part, and these are the pollen grains that you can see through microscope also, magnifying glass also. See, you can see the pollen grains here. You can put out the pollen grains and put on a china dish. So it can be better during the exam time. You put out the pollen grains and put it on the china dish and put some little bit of water, and you can show it to the external. Here also you can dissect the ovary, and if you press the ovary at the end, you will find small ovules. Small ovules. This also you can put on a china dish, and you can see these are the small ovules. Ovaryr bhitore ovules thake. এটাও চাইনা ডিশে অল্প ওয়াটার নিয়ে রেখে এক্সটার্নালকে you can show it to the external. So pollen grains and ovary you can show like this way. Put in the china dish, put some little bit of water, and by using microscope also you can see, using magnifying glass also you can see. So hope you have understood everything. Thank you.